Now, clinical biochemistry. All right. Now, in this first lecture, we will be discussing topics like the use of biochemical tests, the specimens that are collected, how are they collected, what are special considerations that you need to have in mind. And then towards the end, we will look at lab safety and quality control measures, because the sample that arrives at a clinical biochemistry lab, your job is to give a report, and based on that report, the patient's treatment will depend. So it is really important that there is quality check in the lab. The results are really what they are intended to be. All right. So we will look at this. So we will spend a little more time on this particular aspect. All right. Towards the end, we just look at point of care testing just quickly. So basically, whenever there is a patient, all right, and doctor sees the patient and tries to make a diagnosis, there's a diagnostic framework, right? And I think you all must be aware of this. First of all, you take history from the patient to find out what is the problem, since when the patient is having this problem. Getting clues from the history, the doctor will then examine the patient, getting clues on any physical signs that might be there. Okay. After that, the doctor may come to a provisional diagnosis or may shortlist a few diagnoses that he think might be a possible cause of the illness. Then he will order some diagnostic tests to confirm what he thinks is the diagnosis. Now, these diagnostic tests fall into three major categories. These three major categories, okay, you have the laboratory services, which we will now look in more detail later. Could be imaging, X-rays, MRI, CT scan. It could be physiological tests like ECG, EEG, lung function tests. So there's a host of tests that could be ordered in order to reach at a diagnosis. These laboratory services now are subdivided into many different types of services. All right, you have hematology, histopathology, genetics, immunology, microbiology, and then you have the clinical biochemistry here. Okay, those who are taking MLS 300, you guys are over here, histopathology or cell pathology. Now, clinical biochemistry is basically looking at the chemical composition of body fluids. There are three major subdivisions of clinical biochemistry. There is core biochemistry, the tests which are commonly done, and there is specialized tests which are not offered by all labs, but there are special services, special facilities located in certain areas where you have to send the sample for these specialized tests. Okay? And plus there is emergency services, because sometimes you need the result very quickly, because the management of the patient will depend upon the result. So you have these three types of setup of clinical biochemistry labs: core lab, specialized lab, and emergency lab. Okay. Moving on, there are about 400 different tests that a typical clinical biochemistry lab can perform. The scope of our study in this unit is not to cover all those 400 tests. So we will look at actually a group of tests, try to understand their principles and how these tests can help us or inform us in making a proper diagnosis of patient's problem. There are many tests which can be done on automated machines. Nowadays, it's the norm. Most of the tests are done on automated machines. But there are certain tests which can be carried out by using kits which are available, and some analysis are still, they need to be carried out manually. Okay. So as I just mentioned to you earlier, you have core lab facility. This core lab facility is usually present in all hospitals. It is open all the time. Essential and most requested tests are done over here. Highly automated environment. So you have automated machines which will carry out large number of tests in a short period of time. You have special chemistry, specialized tests, and then you have point of care testing, which could be in the CCU, ICU, emergency settings, etc., etc. All right. Core laboratory requires quick turnaround time. And these are the common tests which are done in core laboratory. You don't have to go through the list, which is just there for your reference. Then you have specialized tests which are offered, as I just told you, in special labs, special services there. Okay. There are certain tests which are called dynamic tests. So these kind of tests you can't perform on a single sample. You need multiple samples. One example is glucose tolerance test. Anyone knows what is glucose tolerance test? Anyone has heard this name? Glucose tolerance test or glucose challenge test? You give the patient 50 grams of glucose and then you test blood sugar every half an hour. Okay, so you have to take multiple samples to see how the blood glucose level goes up, how it comes down. There are many other tests where you have to test sample at different time of the day, early morning, late night. For example, cortisol hormone, you have to measure it early morning, 4 o'clock or 5 o'clock in the saliva. So there are certain tests that you have to do at certain times and multiple samples. Okay. Now use of biochemical tests, just building on what I was just telling you. So these tests or these reports can help us in making diagnosis. They can help us in screening populations. You have sometimes you have these tests before you join the university or you get employed by someone. You have these tests, right? Normal screening tests. Prognosis. Now these tests can also tell us whether the treatment patient is receiving is working or not. So if you are doing the tests regularly, you can find out whether the patient is improving or is getting worse. Okay. In addition to this, 2 more applications of clinical biochemistry is research and clinical trials. So many of you, I hope, will not stop at this level. You guys will go on doing your postgraduate degrees and PhDs and so on. So then you will be looking at research and clinical trials and these kind of areas where clinical biochemistry has a very important role to play. Okay. The clinical biochemistry process, now we will be looking at this process during the rest of the lecture now. So first of all, it starts with a clinical question that the doctor asks, what is wrong with this patient? Then he orders, he requests some tests with clinical data, sample is collected, it is sent to the lab, and then it is first checked or recorded in the system with patient's name, patient's ID, so that you don't mix up samples. Okay. Then analysis is done, quality control measures are in place, the data is collected, interpreted, and then the report is given to the doctor, which then completes the whole picture. The jigsaw puzzle is now complete. So the clinical impression that the doctor had is now supplemented by the laboratory data, and the doctor can now with confidence make the diagnosis and treat the patient. Okay, so this is a very important aspect of patient care. Now, how results are expressed? Have you seen any, silly question, you must have seen many clinical reports, right? How the results are expressed? If it is serum calcium level, for example, how is it usually expressed?
Like a number, and that will give you a range.
Like a number
And then
with a unit, and then you have a reference range. So this is a normal range. It should be within this normal range. If it is below or it is high, then the report is abnormal, right? Okay. Now the units which are now internationally in use are the SI units. Okay, I don't know how to pronounce this. Anyone who can help me? Semi, whatever, international D units. Okay, SI units. These SI units are commonly used. There are some non-SI units which have been used so commonly that they are still in use. Okay. So these are the SI units. For length it is meter, for mass it is kilogram, for time it is second, for example. These are derived units. For frequency, hertz, for force it is Newton, etc., etc. And then these are the non-SI units which are in use, although they are not under the SI classification, but they are still in use, like minute in hour, day, etc. Liter in volume. Okay, volume in liter, sorry. And these are the prefixes used with SI units. So you need to be familiar with these. If you see this letter, femtoliters, then you know that this is 10 raised to power minus 15 liters. You know one red blood cell, RBC or red blood cell, the volume of one red blood cell is actually in femtoliters. Okay, so when it is expressed on the report, it will be written as 80 to 100 something femtoliters, FL. So you should know at least what these prefixes mean, okay, when you are interpreting a report. Okay. Now understanding concentration. Now because you are dealing with, in the biochemistry lab, you are dealing with samples which are solutions. Whenever you talk about solution, it has two components, solute and solvent, and concentration is a very important aspect there, right? And the reagents that you are using to do the test, also concentration is very important. So this concept of concentration, for example, in terms of percent solution, molarity, molality, normality, I know these are very dry concepts, but these are important for you to know what these mean. In the second lecture, which is about fluid balance, we will look at these in more detail and try to understand what is the difference between them. Okay. Serial dilution is a very common method used in the labs. If you have a concentrated solution, you can dilute it serially and you will have serial dilutions. Moving on to specimen considerations now. There are the three major specimens that clinical biochemistry will deal with is blood, urine and CSF. These are three major body fluids. But less common specimens also include amniotic fluid, duodenal aspirate, gastric juice, etc., saliva, stool, so on. Okay. Blood is collected using venipuncture or phlebotomy. All right. CSF is collected by spinal tap or lumbar puncture, as it is called sometimes, and there is no special method for urine collection. Right. Easy to collect and plenty. Right. The choice of specimen type depends on what are you looking at, the analyte that you want to measure. If there is something which is present in blood, then of course you have to take blood. But if there is something which is easily assessed in urine, then you don't have to go for blood. You can take a urine sample and assess the substance in there. Okay. CSF is taken in special situations, and of course ease of collection will also determine which type of specimen you will go for. Okay. Anyone knows difference between plasma and serum? Any idea? You may have heard these two terms, I suppose, right? Related to blood. Any difference between the two, or it's just synonymously used, these two terms? Any idea? Anyone? Okay, if you take blood, all right, and put it in a glass tube, what will happen if you leave it there for a few minutes? It will clot. It will clot. You will have a clot at the bottom and you will have a clear fluid on top. Okay. If you do not allow the blood to clot, if you put some sort of anticoagulant in the blood and then you spin it in the centrifuge, all the cells will settle down, you will have the fluid on top. Now this fluid is different from the fluid if you allow the blood to clot. Okay. So if you collect the fluid after blood has clotted, this is called serum. Okay. If you just spin down the blood, you put anticoagulant, you don't let it clot, just spin down the cells, take the fluid portion, it is plasma. So plasma contains clotting factors because they are not being used up. It contains platelets, okay, to some extent, but mainly the factors. Platelets would have settled down. Okay. Serum is similar to plasma without clotting factors, because clotting factors have been used up in forming the clot. Right? Okay. Sometimes if you have a small wound on your hand and a scab forms on it, okay, sometimes you see a very clear fluid around the scab. That is serum. That is without the clotting factors, because clotting factors have been used up in forming the clot. Okay. So this is the difference between plasma and serum. Now, when you collected the sample, sometimes it needs processing. Sometimes it needs immediate processing, sometimes you can freeze the sample and do the processing later. So depending upon what you are looking at, you have to decide which type of sample processing you will carry out. Unless you are doing a whole blood analysis, the sample is immediately centrifuged, serum or plasma should be separated and put in separate vials. Okay. If you are not going to analyze the sample immediately, okay, you can store certain samples at 4 degrees up till 8 hours, or there are some samples which can even be frozen at minus 20 degrees, can be analyzed later. All right. It will depend upon which test you are going to perform. Okay. Then there are many other variables that you have to consider, like patient preparation. There are any problems in collection, transportation, processing, storage, any of these, the chain of events. If there is any problem anywhere, for example, the storage is not at proper temperature, or the time taken is longer than the time allowed for that specimen to reach the lab, all these factors can affect the quality of your report, and it will not reflect what the report is supposed to reflect. So good communication with all personnel involved helps ensure that whatever plans are in place, they meet the needs of the laboratory and ultimately the patient and the physician. Okay. Coming on to now lab safety and quality control. Any question until now? Actually, it's quite general what I've been telling you. So I think you all know all these facts, just reiterating the same facts again. Safety hazards. Now, when you see this mark over here, it means I want you to ask me something or tell me something. Okay. So can you just try to list common hazards that you think are present in a typical clinical biochemistry lab? What type of risks do you think are there?
Chemical spills.
Chemical spills, broken glass,
Flammable fluids.
flammable fluids,
Pathogens.
sorry?
Pathogens
Pathogens.
in the blood, in the sample.
Yeah, okay. So you are dealing with infectious specimen, for example, right? So there are a lot of hazards which are in a typical lab. Okay, this is just to list a few. All right, and if you look at the various checks and balances which are in place, most countries have an Occupational Safety Health Act in place. So you have to follow certain guidelines when you're designing a lab, when you're putting stuff there. Everything should be considered. Awareness and education among people who are working there is very, very important. And last but not the least, personal protective equipment. Okay. So if you go back to our minion, he's safe. He's wearing goggles, gloves, and closed-toe shoes, right? At least the minimum. Okay. But accidents do occur despite all these safety measures. So we have to be very careful when working in such environment where you are surrounded by so many different types of hazards. Okay, just some examples. Okay, and if it happens in a university, the teachers are fired. Okay, so have to be careful. Now, these hazards can be broadly divided into 4 groups. Has any one of you gone through any safety courses, biological, chemical, radiation? Not yet? Okay, but you all will have to before you start working in a lab. Looking at biological safety, one thing very important. Whenever you are dealing with blood or body fluids, okay, they should be presumed infectious. Even if you know the patient is absolutely healthy, you should deal with this specimen as potentially infectious. Okay? You know what is the most common injury that occurs when you are collecting blood sample from a patient's vein? The most common injury that occurs? Or when it occurs? Needle stick. Okay, needle stick is the most common injury that occurs. And when does it occur? So you had a needle, you took off the plastic cap, you insert the needle in the patient's vein, you collected sample very nicely, then what you do after that? You take the needle out, then you recap the needle, right? You never recap the needle, okay? The most injuries that occur is while recapping the needle. So, into your thumb or your finger, right? So, you never recap the needle. The safety technique is never use two hands to recap the needle. You can use either one hand, the cap is on the table, and you just go after the cap. You take the cap, and with one hand, you just click it close. Nowadays, they don't even recommend this. You have a sharps bin next to you, you collect the sample, you transfer it into whatever tube you want, and the whole needle you throw into the sharps bin. Don't try to recap it even. So, recapping has been the most common cause of needle stick injuries, has led to many people getting hepatitis and AIDS and whatnot. Okay, so I cannot stress this more than this. This recapping is the most dangerous thing you will do when you are collecting blood samples. Never try that. Okay. Gloves, gowns, and face protection is very important. Hand washing, hand washing, hand washing. Never underestimate the power of hand washing. Okay, and the hand gels, these soaps, they do not replace a proper hand wash. So regularly, proper hand washing has to be done. Okay. Chemical safety. Now, chemicals in biochemistry lab, chemicals are everywhere. Okay, you will be sitting, sleeping, and drinking chemicals. All right. So you have to be very, very cautious about chemicals. All the chemicals in the laboratory must be labeled very clearly, and they should have what is known as MSDS or SDS, safety data sheets, or material safety data sheets. MSDS should be available. This SDS will contain basic information about the chemical, what risks it poses, what are the first aid measures if somebody is exposed to it. So all this information will be there. This MSDS or SDS should be available for all the chemicals which are in the lab. Okay. Then all employees must be educated on how to interpret chemical labels. There are certain color codes, certain symbols, everyone should be familiar with those. Okay. Health hazard, health warning labels should be maintained, and chemical spill kit should always be available in case there is a spill. Radiation safety, not that common, but there are certain assays where we use radioactive isotopes. So if you, in your laboratory there is some radioactive work going on, you have to be very careful dealing with the radiation in the lab. Okay, there must be regular and systematic monitoring, and record must be maintained of how much material has come into the lab, how much has been disposed of. So you know exactly how much is presently housed in the lab. Okay. Now radiation doesn't create Teenage Mutant Ninja Turtles or these kind of big, it's all myth. Okay. In addition to ionizing radiation, the harmless non-ionizing radiation can also be a health risk, and in the lab there are many sources of such types of radiation, energy beam microwaves, heat lamps, lasers. So all these, although it is not that injurious as ionizing radiation, but it can also pose health risk. So has to be carefully monitored. Okay. Fire safety. A clinical biochemistry lab has all the ingredients for fire. Okay. So you should not let them mix together, otherwise kaboom. Okay. So fire safety is also very, very important. Okay. And you should know different fire extinguishers, right? Do not spray water on electric fire, you know that, right? Okay. So you have to know which fire, how to control, how to extinguish that particular type of fire. All right. Do you want to have a short break? And if you have any questions, I can answer those before I go to the next topic.